hear me?
Yes. Can you see my presentation in full screen?
Yes, yes. It's not in. It froze my. Oh, now there you go. There you go. Yeah.
Okay. Great. Great.
Yeah. See that. Let's see if I can. All right. I think we can get started, and whenever the people are arriving, I will admit them in. Well, Maor, thank you very much for accepting our invitation to be here and to talk to our students and our visitor on this webinar. I really appreciate. I will. Dr. Maor Matzrafi is from Israel, and he will introduce himself a little bit more. But he will going to speak about climate change and how it exacerbates the damage of weeds through reduced herbicide effects. Or the floor is yours. Thank you very much for accepting our invitation, and looking forward to hear you.
Thank you very much, Luis. It's a pleasure to be here. I think that you know, in times like this, when we have all of this pandemic outside, the fact that we can gather together and have some seminars and webinars online, it's one of the least advantages that we have for that. And as you said, I'm going to speak about climate change and weed science today. My lab and my research is currently in the VR Research Center. We are part of the Agricultural Research Organization in Volcani Institute. And this is the parallel for the USDA, for example, in the US, and INRA in France, and other authorities that I'm sure that you guys have in your different countries. I'm going to try today to get your heads wrapped around this whole definition of climate change and weed management, especially about herbicide efficacy. And there's going to be some, you know, issues that are maybe more familiar to some of you and maybe less to others. Please feel free to ask questions during my talk using the chat. You can write the questions. I can see the chat is working, and then I'm going to try and answer most of the questions according to, of course, what you would like to know. So let's get started. If we're talking about the impacts of climate change, and I'm sure that most of you heard about some of them at least, we're talking about an increase in temperatures in different areas around the world, different regions. We're going to have an increase of approximately 5 degrees Celsius. In some places it's going to be less, some places it's going to be more. Here on the right side, you can see that this clock of extreme weather, extreme temperature event is running from the start that you saw in the early, I'm going to put it back, the early 1900s until 2000. We can see that there is an increase in the extreme weather event. This is also something that is very, very important for agricultural production. And I'm going to talk a little bit more about it in the rest of my talk. The CO2 levels are also rising. We have a lot of different issues that are related to CO2, especially if we talk about the difference between C3 and C4 plants, weeds, and crops. If we talk about food production, we know that there's going to be an increase, 6 to 16%, depends on the crop. Crops that are more plastic and can adapt, or that have been bred for this kind of characters, more efficient under increased climate change, but still there are crops that are still adjusting to this, and we know that there is a lot of work on that in the breeder side. The current yield loss that we have from weeds is approximately 34%. I'm sure that most of you know the paper by Orke from 2000 and something. But we know that under climate change, we're going to see more yield loss that is related to extreme weather events, like high temperature and fluctuation in precipitation. And these issues are very important for us also in weed science, because some of our weeds are more adaptive than our crops. And this is why they are going to behave differently or adapt differently to climate change than our crops. And this is one of the dangers that we have today. If we're going to look at a specific case, and this is something that I brought back from home, from Israel, and here you can see we have different extreme weather events across the different decades that we have from the 70s of the previous century until 2020 almost today. And what we can see here in those two figures, we can see the extreme weather event. We have calculated all the temperatures that are higher than 26 a day and 15 at night. And when we have blue dot, it means that this was an event of 1 day. 2 dots, the orange dot means that we have 2 days, and the gray dots means that we have 3 days. So we have an increase in the number of events of extreme weather that is over 26 a day and 15 night temperature. Of course, this is Celsius temperature. And what we can see between those two different distinct areas in Israel, as you can see in the map, Dorot is in the southern side of Israel, and Kfar Yoshua is in the northern side of Israel. These two areas are being used for growing wheat and other crops. They are very, very, very important agriculturally in Israel. And we can see that in both of them, we can see some kind of an increase in the number of extreme weather events, mostly for the 1 day. Here there was some kind of an abnormality between 2000 and 2009, but also for 2 days and 3 days events that are increasing in the last 10 or 20 years. When we have these kind of events at the beginning season of wheat growth, we can expect that there's going to be a response from crop and the weed. And these are the things that we are trying to analyze the response to, and the stuff that we are worried from. If we're going to look more precisely into November at the end of the month, this is the month where we sow our wheat in Israel, and when we start applying herbicides and other pesticides in the field. And we can see that the temperatures, even though this month is categorized as autumn or at the beginning of winter, we have very high temperatures in some of the days. Here we recorded in 2021, 2 days with high extreme temperature of almost 35 degrees. May I remind you that most of the temperatures that we have in this season are less than 25. You can see the other days are less than 25 degrees. And this abnormality can cause different things that will reduce the efficacy of herbicides, that will make the weeds grow faster, or will change the activity of the crop and the reaction that it has for the treatments that we are doing in the field. If we're talking about crops, and I've reviewed some of these papers that you see over here, we have a lot of research that is related to the response of different crops to climate change, as you can see over here, papers from 2017 and 2020 and others, papers that are dealing with wheat, some of them are dealing with maize, soybean. And when we look at these different topics that people have researched, we can see that they are going from response to ozone and CO2, resilient to drought and other issues like salinity. All these issues can be categorized as climate change actually. And the research is very advanced in the field of crop science. Unfortunately for us in wheat science, we have less research related to that. I'll show you some papers that are dated back to the 90s, but there is a lot of gap between the work that we need to do and the work that we have. The data that we have is still not sufficient in order to predict climate change and how we can cope with it, how we can manage our weeds better. So if we talk about weed management under climate change, I'm going to show you some examples. And first of all, we want to talk about what are we studying? What are the issues that are most important to us, and how do we analyze them? How do we approach these issues? And if we look at the specific issues, this is something that I took both from Louis Ziska's paper. I'm sure some of you know him and read his papers, and another review, an opinion paper that I did a couple of years ago. And generally, we're talking about 4 specific or 4 main subjects. The first subject is related to phenological shifts, weed distribution, demography, fitness relative to the cost, to the crop system. And these issues are related to changes that we think that weeds can get under climate change and will somehow interrupt with our weed management. Now, this can be ecological shifts, This can be biological shifts, morphological shifts. Weeds can change the time of flowering. They can change the time of senescence. They can change their growth relative rate. And this is how they can affect the competition with the crop. The second thing that we have that is very important is shifting the efficacy of biological control. I'll show some examples, but there are a lot of studies that are related to biological control using insects that are showing that with climate change, there is also a change in the efficacy of the biological agents. Some of our insects will behave differently. Some of them are not used to high temperatures. Some of them are less efficient at high temperatures. Their eating capacity is less at high temperatures. And this can affect their ability to control the weeds and to actually do their work as biological control agents. The third thing that I'm overviewing here is the phenotypic plasticity or epigenetic changes. And these papers are very rare. I found only 2 or 3 papers that are related to the epigenetic changes. This is a subject that is really very, very new. If we're looking at climate change and epigenetic changes that are related to weeds, how they will react to different evolution that is related to climate change. We know that there is some kind of an effect that can have that the material in the plant can have, and then this effect can be translated into a reaction in the progeny generation. I'll show you some examples from that, but this is something that is very important. If there is a stress that the plant is experiencing, the offspring would probably be more resistant or resilient to this stress. This is something that we also need to know and investigate further on. But the main thing that I'm going to talk about today, it's like it's been hinted, in the name of the presentation, is mainly the differential herbicide response under change environmental conditions. We have several issues that are related to conditional resistance. This is the way that we categorize the different response that we have from weeds under different environmental conditions. And I'm going to show you some examples for the research that have been done in the last few years. So first of all, if we talked a little bit about differences in growth and things that are related to environmental changes, here is a very nice example from the 1990s. Louis Ziska and Bounce did some really nice work, and they looked at the growth and other aspects of biomass that are related to the changes in CO2 levels. Here you can see that they have different weeds and different crops, and they compare between them. On the left side, it's the percentage stimulation of plant biomass, and on the right side, it's the percent stimulation of CO2 assimilation. And we can see that in almost all crops versus weeds, we can see that there is an advantage to the weed over the crop that we compare them to. There are different crops here like maize, sorghum, and other different families of crops. We can see that amaranthus species are doing very well even under high elevated CO2. They're doing well more than what they would do under low CO2. Also the Echinocloa species and Setaria species. And we also have like more CO2 assimilation for some of these species. And generally, you can see that the competition between weeds and crops can be more severe under increased CO2 levels. Of course, at this moment, we have to distinguish between C4 and C3 species, amaranthus species and Echinocloa species, or C4 species, some of the other grass weeds that we see here. And this can give them also an advantage, but we can see the same thing also for other C3 species in different studies. The second study that I want to show you is related to the growth of Parthenium hysterophorus under elevated CO2 levels. Parthenium is a very, very interesting weed. It has invaded more than 50 countries around the world. It produces a lot of specific allelopathic compounds. Some of them are harmful for men, some of them are harmful for animals. And the damage that can be caused by this specific weed is very high. We can see here the difference in plant height and dry biomass and other different characteristics, such as number of flowers, and number of leaves between plants that were grown under ambient CO2 levels, 400 ppm, and elevated CO2 levels, 700 ppm. This work was done in Australia by Ali Bajwa and his collaborators. And we can see that for each of the different characteristic or parameters that we see over here, there is an advantage for plants that are grown under high CO2 levels. Now, this advantage, as I said before, can be translated into higher competitive abilities versus the crops that are growing within at the same field. The case of biological control is very interesting because there are not a lot of works that are related to weeds. This is a work that I actually found from Insect Science. It was published a year ago, and it's related to a moth, Plutella, and it's a parasitoid that is used as a biological control agent, a parasitic wasp that is actually controlling this moth. And here you can see the differences in the parameters that we have from different temperatures. When we increase the temperature where the moth is growing and the wasp is growing, we can see the effect that it has on the number of eggs and larvae, and also on the number of pupa and the pupation rate. And we can see that when we increase the temperature, there is a reduction in the number of eggs and larvae and also the pupas. And that means that the specific biological control agent that we have can have a problem in reproducing an overall weed control, or in this case, insect control that we wanted to have. Now, if you look at this figure, it summarizes it very nicely. The performance is on the y-axis and on the temperature is on the x-axis. And we can see that from a specific temperature, we can see that there is a very very high slope, a very dense slope of the number of parasitoid wasps compared to the host. For an example, at 25 degrees, you can see that the host has a lot of individuals and the performance is very high compared to the wasp. In other temperatures, it was very similar. We can still see that, for an example, at this scenario, the parasitic wasp can still cope with the host, with the moth. In this one, also, it's even more closer in of performance. But when we look at 25 degrees, we can see that the host is really running away from the parasitoid wasp. And this issue can create loss of efficacy in the biological control. There was a nice study that was done as a meta-analysis, and this is from 2007. It was published in Global Change Biology. There are a lot of really nice papers that are related to climate change and the effect on weed management and also other management of other pests in Global Change Biology. If you're interested, I think that it's a really good journal for you to find some papers in there. And here you can see the meta-analysis that they did, and they tried to find the herbivore response to elevated CO2. Now, what you see over here in this figure is the proportion change from ambient CO2. That means the elevated CO2 and the effect that was measured. Now, I took several effects that I think that are mostly important, and you can see them over here. First, the abundancy, the total number of individuals, the relative growth rate of these individuals, the amount of food ingest, how much do they eat, because the more they eat, the more they are efficient as biological control agent, the number of days necessary to complete development, and relative consumption rate. Also, this is some kind of a relative measurement for how much they eat. So if we look at these three parameters that I've marked with the red arrows, the abundancy was reduced. And I want to remind you that this is all insect. This is a meta-analysis. So we can see the general abundancy of these insects was reduced with an increase of CO2. There is also the relative growth rate and relative consumption rate. And these parameters are very important when we want to have efficient biological agents that can reduce the amount of weeds or other pests that we have in our field. When the number is reducing, it means that they are less efficient. So this is something that will probably affect the whole mechanism or the whole system of integrated pest management. And it's gonna reduce the crops the crop yield, and even the crop quality that we have afterwards. So if we talked a little bit about those general subjects, and now I want to focus my lecture about the differences in herbicide response under changed environmental conditions, what I defined earlier as conditional resistance. We have different cases of this conditional resistance, and we always try to ask ourselves, what is the mechanism of this conditional resistance, in order to understand how we can prevent this detoxification or those reduced herbicide efficacy consequences. We need to understand what are the mechanisms that are involved in the system. Now, this is a table that I've prepared, and it's concentrating most of the cases that we had until 2019 that are related to response under environmental changes. And you can see here different responses. You can see here different weed species, and these are the active ingredients. We have the R and S. This is either resistant or susceptible populations that were tested. And we have the environmental factor, and of course the citation. If you want, you can go and read those papers more thoroughly if you are looking for more information. So we can see, for an example, this is a specific case of response to dichlorphosmethyl. This is an ACCase inhibitor. This is something that we checked in 2016, and you can see here that there are different weed species that we tested for that. Some of them were resistant, some of them were sensitive. And what we see in general is that there is a reduced efficacy of the herbicide under elevated temperatures. Now, we tested them in 10 versus 16, and in 10, 16 night day versus 28, 34 night day, and we saw reduced efficacy to dichlorphosmethyl. There are other weed species, for an example, the Ordeon glaucum in response to paraquat. And this is a unique case also, because here you can see that there is a different response between the sensitive biotype or population to the resistant biotype. While the resistant biotype has reduced efficacy at 15 degrees Celsius compared to 30 degrees, the sensitive biotype has reduced efficacy at 30 degrees compared to 15 degrees. It means the resistant and sensitive accessions or populations have a different response for temperature elevation and to paraquat. And this is something that you can see in some of the population. It's mainly related to the mechanism of resistance, but I'll talk a little bit about this in some of the next slides that I will show, so I don't want to talk about it too much here. Another thing that we can see, for an example, is the response to glyphosate. And here we can see different species. We can see Coniza species versus Ambrosia species. We can see that there is a reduced efficacy under elevated temperature for the Coniza species. But for the Ambrosia species, we can see reduced efficacy under decreased temperature. So here we also see that species can play a role in how herbicide detoxification works between different temperatures. While for Coniza canadensis or Coniza bonariensis, the response was reduced sensitivity under elevated temperature. For the Ambrosia species, we saw reduced sensitivity under the decreased temperature. So this is something that is very interesting to see, and I think that this is part of why we have to test a lot of weed species and a lot of herbicides, because the response can be so different. Until 2019, most of the studies were on temperatures. Some of the studies that, for example, Louis Ziska did were on CO2 levels, but we lacked studies that are combining both factors together. Now, if we look at the future climate change, we know that we cannot separate temperatures from CO2 levels. These two factors go hand by hand, and they can be combined in order to create specific combinations of climate change, temperature, and CO2 shifts that we would have to cope with, our crops and our weeds also. So this is why we decided to do a very robust experiment that will show how plants will respond to glyphosate under high CO2 levels, high temperatures, and the combination of both factors. And what we had is we took Chenopodium album and Coniza canadensis, and those were two sensitive populations, and we saw that there is a reduced efficacy when we combine them together, or even when we look at those two separately. So this is something that I'm going to show more about the study in the next few slides, but I just wanted to show you that generally we don't have a lot of studies that are working on combined parameters, and this is something that we have to start and see. We saw several new studies from Neil de Burgos with collaboration with Luis also in Luis Zisca and other people from Brazil, but we still don't have a lot of information, or not enough information, in order to assess the combined response of these two parameters. I saw a question. So Esra is, I will say like very shortly that I will talk about genetic basis for conditional resistance in the next few slides. So please bear with me, I'll get to that. If we talk about herbicide resistant mechanism, I'm sure that most of you know them. Maybe most of you even know this paper. This is a very good paper for everybody that wants to know more about mechanism of herbicide resistant, the evolution of herbicide resistant. It was published in 2013. Christophe Delier, a researcher from INRA from France, with Marie Jessenyuk and Valérie Delacour, they did a very good review about all the mechanisms of resistance and genetic behind those mechanisms of resistance. And I recommend this paper to everybody who's trying to wrap their hands around all this subject. This is a very good introductory paper to this issue. What we have generally, and this is something that I wanted to show you, within those mechanisms of resistance and those categories and the definitions, we have three mechanisms of resistance that are related to detoxification and non-target site resistance issues. These are the main mechanisms of resistance that we see when we look on the response to differential temperatures and CO2 levels. And these are the main mechanisms that I'm going to talk about in the next few slides. So this is some kind of an elaboration for the experiment that I showed before. I wanted to show you the results in a more visual way. Here you can see the effect of temperature on different grass weed species in response to diclofop. We used, as I said, Alopeculus, Avena, Lolium, Setaria, and you can see the control is Lolium rigidum that has target site resistance, as you can see over here, which means that it shouldn't be affected by the temperature and either by the treatment. As you can see, the treatment was with the recommended rate of diclofop-methyl. This is a herbicide that is used to control grass weeds in wheat. We can see the control treatment versus the herbicide, and we can see that the response when we are using low temperatures of 16-day, 10-night is very obvious. All the plants from all the population, except from the target site resistant population, have been controlled very nicely. When we move to the next step and did the experiment under high temperatures, we can see that the number of plants that have been survived the treatment and the general percentage of survivor for each population was very high. Some of the survivors, some of the population have reached a survival percentage of 80%. This was very interesting to us. The change in temperature, the shift in temperature, have probably increased their ability to resist or to stand the herbicide treatment. When we looked at those different treatment that I showed you before, we started thinking what's going to be the response if we take target site resistant population versus non-target site versus sensitive population. And we wanted to see if there is a difference that it is also related to the mechanism of resistance. And in here you can see three Lolium populations. One, the KY is very sensitive. The EMR has non-target site resistance that is related to metabolism of ACCase inhibitors. And the NO is the population that you saw before. It has target site resistance. We use diclofop-methyl, and the first part of the slide that I'm showing is the low temperature. As you can see, there is a very good control for the sensitive population, also for the non-target site resistant population, but the resistant population that has target site resistant looks like it's not really affected by the herbicide. Now, if we look, I'm sorry, this is the lower numbers are showing, the one is the X. This is the recommended field rate that we used, and this is 4 of the recommended field rate. Now, if we look at the response under elevated temperature, and as you remember from the previous slide, there was less efficacy for the dichlorphor under elevated temperature. Here we see the same trend, but we can see that there is a difference between the population that is sensitive to the herbicide and the one that has non-target site resistance. The sensitive population can survive the recommended field rate, and some of the plants can even survive twice the recommended field rate. But in the non-target site resistant population, suddenly from a population that has almost no survivors in 1x, we get to a population that has almost 100% survivors in 4 times the recommended field rate. That means that we actually, what we see over here is that there is an increase in the effect in the amount of metabolism through the temperatures that is affecting the amount of herbicide that the population can absorb and can detoxify during this time. And this is a difference that we can see between those two populations. There is probably some kind of an adaptive trait that get the non-target site population to be more resistant, but we see that there is some kind of an increased resistance or reduced sensitivity also for the sensitive population. So after we saw that, we started thinking, what is the mechanism behind it? And at this point, it was very complicated to work with species like Lolium or Avena or Anopicurus, because this species has a lot of genetic variation within the population, the field population. And we decided to switch and move working with some kind of a model species in the grassweed family, in the Poaceae family, Brachypodium hybridum. And we wanted to see what's going to be the response of this species to herbicide application under different temperatures. But instead of working with only temperatures, we also wanted to see what's the time that it takes for the herbicide detoxification during the exposure or after the exposure of the plant to the herbicide. Now, this experiment is a little bit complicated, so bear with me for a second and I'll try to explain it. What we did is we took Brachypodium plants, we grew them till they got to the age of 2 leaves. And then what we did is we sprayed them with the herbicide. And we had 2 different rooms with 2 different temperatures. The low temperature 1610, as I showed before, and the high temperature 3428. And what we did is, after spraying the plants, we started moving them hours after spraying between those 2 rooms. We moved plants from the hot room to the cold room and from the cold room to the hot room. We did have some kind of a control, as you can see over here. The plants that are controlled C hot, they were plants that were not sprayed, and we grew them in the hot room from the beginning until the end of the experiment. And P hot were the plants that were sprayed with the herbicide and grown in the hot room from the beginning until the end of the experiment. The rest of the plants were moved between the rooms. And what we see when we compare both rooms, first of all, we see that if we look at our 2 controls, plants that were treated and grown under high temperature, you can see that they look very nicely, they survive pretty good, and they look almost as a control. Plants that were grown and sprayed under cold temperature, you can see that versus the control, we had a very good efficacy of the herbicide. Now, if we took plants from the cold room and move it to the hot room, we can see that if we move it 2 hours after application and 24 hours after application, even 48 hours after application, plants tend to survive the treatment. And if we move them 72 and 96 hours after treatment, you can see that they are controlled pretty nicely. Now, the same mirror picture can be shown in the other slide. If we move plants from the hot room to the cold room to 24 and even 48 hours after application, we can see that they look very poor and they haven't survived the treatment 21 days. This is 21 days after application. But if we move them only after 72 hours or 90 or 96 hours, they look very nicely. So it means that there is a time frame that the herbicide needs in order to move through the plant and do its action. But if you move it before that, the plant can actually metabolize the herbicide very rapidly, and then the control efficacy is reduced. Now we have here some of these results that you can see the statistics for them, and if you want, you can, of course, you can go and read the paper and get more data about this experiment. Now, after we saw that, we started thinking, okay, we want to see what is the detoxification pattern, and we want to see the actual mechanism of detoxification under high temperature for the brachypodium that we tested for pinoxidine. Here you can see an illustration of how pinoxidine is getting detoxificated through the different stages. For those of you who doesn't know, pinoxidine is actually a parent molecule. It's not an active ingredient per se. It's not an active molecule. When it penetrates the plant, it goes through some kind of a spontaneous hydrolyzation, and then you get the active molecule. As you can see here, the N2. This is an inactive molecule. In the path of detoxification, we have several inactive metabolites that can go back to being active metabolites or can stay inactive metabolite. The M4 is actually the first detoxification pattern that we see, and here you can see this group OH. I'm sorry, there is something with a microphone that is open. If you can please mute, it's going to be very nice. So we can see the M4 over here, the group that you see over here, the OH is where the sugar, the glucose is being conjugated at the end, and this is inactive herbicide. Now, in some of the plants, the form of M4 is also going to be an inactive herbicide, and this is going to be the end of the metabolism pathway. So if we look on our two brachypodium accessions, and here we have one brachypodium accession that is sensitive to pinoxidine, and the other one that is resistant to pinoxidine. We grew them under two different temperatures and treated them with pinoxidine. And then we tested to see the amount of metabolites for 2, 48, and 26 hours after herbicide application. What you see over here is three metabolites, three main metabolites that I'm going to show you. The inactive molecule, the parent molecule, the active molecule, the N2, this is the actual herbicide, as I said, and this is the intermediate metabolite that is most probably not active as a herbicide. What we can see here is we can see a decrease in the amount of the parent molecule through time, and it's generally pretty similar for both accessions, but you can see that under low temperature, the decrease in the amount of the active molecule is lower. It takes more time to detoxificate the active molecule under low temperatures. The same pattern was shown for the active molecule, as you can see over here. The statistical significance tends to show you that under low temperature, we have more pinoxidine, more active molecule than under low temperature. There is a difference between the resistant and the sensitive accession, but I'm not going to go through it. I want to show you mainly the response to temperature, and this is the important thing. And when we look at the last slide and the last part of the figure, we can see the M4, the intermediate metabolite, as I showed you, that's going to be conjugated to glucose. And here, this is the interesting story, because if we look at the amount of this inactive metabolite, we can see that the accumulation after 2 hours only under high temperature for the resistant biotype was very high, and we haven't seen any of this inactive metabolite 2 hours after application in the sensitive biotype. Now, there is an increase in the sensitive biotype 48 hours after application, but it's most probably too late for the plant to cope with the herbicide. It suffered from a very severe damage, and even if we have high detoxification ratio, it would probably not affect the activity of the herbicide 48 hours after application. Now, if we look at 48 hours after application for the resistant biotype, we see some kind of the same trend that we saw before. There is more of the intermediate metabolite for this part, but there is an increase in the intermediate metabolite also for the low temperature. And that means that most probably The resistant biotype, the fact is that it has some kind of a response to the temperature, but the basic response that it has to the herbicide is better than the sensitive biotype. This is some kind of a quantitative response, and I'll show a little bit more about it in the next few slides. But generally, what we have to understand is those two mechanisms can work under the same detoxification pattern. They can use the same enzyme, but maybe the resistant biotype has a general starting point that is higher by the amount of herbicide detoxification than the sensitive biotype. This is something that we have to measure in the level of the enzymes, the cytochrome P450 or other glutathione transferase or glyphosate transferase that are working here. We have to measure it in order to show it, but this is a very good clue for the difference between detoxification pattern of these two accessions. The next thing that we did is we tried to look at the metabolic resistant related genes, and this transcriptome analysis was done 24 hours after herbicide application. We have here the different treatments. X is using only pinoxidine under low temperatures. H plus X is using pinoxidine under high temperature, growing the plants under high temperature. And only X is another control that we have where we grew plants under high temperature without the use of the herbicide. Now, what we can see over here is 3 different gene families, cytochrome P450s, some of the GSTs that we saw, and UDP glycosate transferase that we saw. This is the part that these are the genes that are related to the detoxification pattern where the glucose is being binded to the intermediate metabolite. And this is a very interesting thing, what we found. If we look at these 2 cytochrome P450 or even 3, the CYP72A, this family is known as part of detoxification in several other studies. It was found in Lolium rigidum and in other grassweed species to be related to detoxification of ACCase inhibitors. And what we can see over here is that the increase in the expression of these genes is actually not so different between plants that have been treated with pinoxidine only and plants that have been under the combination of high temperature and treatment with pinoxidine. Now, under high temperature, we haven't seen this increase, so it would probably mean that the pinoxidine is triggering the increase in the cytochrome P450 expression. Now, the same thing was also for GST and also for the other family, the UDP glucose. When we look at these results, we probably think, okay, so we can see that under pinoxidine with low temperature and pinoxidine with high temperature, we see the same expression. So if we see the same expression, what does it mean? Why does the herbicide react less under low temperature and not as the same as high temperature? And this is something that we also thought about for a lot of time, and we try to understand, okay, so there is no difference in gene expression. And then we started looking on other genes that were differentially expressed in this experiment. And what we saw is that there is another group of genes that has been elevated. And these genes are peroxidase genes, and they are related to detoxification processes, mainly to scavenging of reactive oxygen species that are part of detoxification processes. In the process of cytochrome P450 activity, there are a lot of reactive oxygen species that are produced during the process. When they are produced, they need to be, someone needs to take care of them. And this is the work of peroxidase. Now we can see that the increase in peroxidase happens only under the combination of high temperature and pinoxidine application. And when we looked at the accumulation of reactive oxygen species, we saw a very interesting picture. Here we have hours after treatment, 2, 8, and 24 hours after treatment. And what you see is the color, the staining color, the GFP color that is marking the elevation in reactive oxygen species. Now we can see that after 2 hours, we have high level of reactive oxygen species in almost all treatments, high temperature and pinoxidine and the combination. But when we look at 24 hours after treatment, we can see that under the combination of high temperature and pinoxidine, we can see that there was a very slight amount of these reactive oxygen species. We also have statistically significant results for that. If you want to look at the paper, you can see it later. But you can see the difference between pinoxidine treatment alone and the combination with high temperatures. This is a very distinct difference. That can be a hint for the fact that probably both situations has an increase, or the accession that we are working with has an increase in the expression of cytochrome P450 in both situations. But the activity of the cytochrome P450 is only enhanced under high temperatures. And this is why we see resistance that is increasing under high temperatures. And as I said before, this is only a hint. We still have to figure out the whole mechanism. We still have to look more into the related genes and proteins that are working. But generally, this is some kind of hypothesis that we produced during this research. There were other researchers that were working about temperature-dependent response, and here is another research that came from Mitila Jugulam's lab. If you're looking on good papers that are related to climate change and herbicide response, Mitila's lab did a lot of work on that, very interesting work. And here we can see the response of Palmer amaranth, Amaranthus palmeri, to mesotrione, an HPPD inhibitor. Here we have the low temperature. This is 1525. You can see the untreated versus the untreated plants, recommended field rate, and you can see that they have been controlled very nicely. When we go through the temperature to the increasing temperatures, we can see that in the high temperature scenario, even under twice of this dose, there was no control of the weed. The response of the weed to the herbicide is decreasing, the sensitivity is decreasing as the temperature is increasing. And this is something that I showed you before, and it's very interesting to see it in other weed species. Now, for the mechanism that dominates this phenomenon, here you can see those researchers, Amar Godard, that worked for Mithila at that time, inspected the absorption of the herbicide and the translocation of the herbicide. And you can see the high temperature versus the optimal temperature and the low temperature. These are the different colors that we have for the lines. Here we can see that, for example, for the absorption, we had high absorption for the high temperature. As you can see, it's statistically significant in most time points from the two other temperatures. But if we look at the translocation, we can see that the translocation, the TL is the treated leaf, ATL above treated leaf, and BTL below treated leaf, when we divide the plant during the translocation assays. And here we can see that if we look at the compounds in the treated leaf, we saw that in both low temperature and high temperature, most of the compounds stay in the treated leaf. In the medium temperature, some of the compounds leave the treated leaf. If we look at the compounds in above treated leaf, we can see that most of the compound, the C14 compound, stay in the treated leaf, okay? Or we don't see it in the above treated leaf in the high temperature, and the same thing we see for the compounds below treated leaf. So generally, what we can assume from this study is that when you spray Amaranthus palmeri with mesotrione under high temperature, the absorption is okay. It will be absorbed into the leaf, but the translocation is impaired and the herbicide is not moving as you would want it to move. And this is why herbicide cannot act and cannot control the plant as effectively as it can in low temperatures. The second case that I want to show about reduced efficacy that is also from Mithila's lab is reduced efficacy under increasing temperature in Basilia Scoparia. The Basilia Scoparia is a very interesting weed. We have a lot of studies that are related to that in the US mainly, but it's also spread worldwide. We have it also in Israel. And here we can see two different herbicides that they worked with, glyphosate and dicamba. And what they saw is that there was a decreased efficacy for both herbicides when they treated them under high temperatures. And when they wanted to investigate the mechanism, they tried to look first at the absorption, as I showed before for Palmer amaranth. So this is the absorption rate under 3 different temperatures, as you can see. The T3 is the high temperature, T2 is the medium, and T1 is the low temperature for both different herbicides. And you can see that for glyphosate, they have found that there is a reduced absorption under high temperature compared to the low and the medium temperature. But for dicamba, they saw that the absorption is almost the same between all three temperature regimes. The second thing that they looked on is the translocation of the herbicide. And here, as I showed you before in the other graph, they looked at the treated leaf and above treated leaf and below treated leaf. And for the glyphosate, they haven't seen any differences. So they have concluded that the absorption is the issue with glyphosate because there is no difference in translocation, not statistically significant for this instance. But for dicamba, they saw something very different. They saw that the translocation of the C14 compound was disrupted, and they saw that most of the compound stayed in the treated leaf in comparison to the low and the medium temperature. They saw that in the high temperature, most of the compound stayed in the treated leaf, and they have deducted that the mechanism of resistance is reduced translocation. Now, what we saw here is we saw two different weed species. We saw them with 3 different herbicides, and we saw that the mechanism can be related to absorption and can be related to translocation, and the differences between those weed species would probably govern some of these issues that are related to the mechanism of resistance. And this is why, as I said before, we have to test as many weeds as we can to as many herbicides as we can in order to find out these issues. Now, maybe the last mechanism versus phenotype case that I want to show is related to plant response to glyphosate under different environmental conditions. And this is the study that I've mentioned before. We wanted to test the response not only to temperature, but also to high CO2 or low CO2 levels, and in combination of both factors. So what we did is we used environmentally controlled chambers, and we had 2 different temperatures, 32, 26 day night and 18, 12, and we had 2 CO2 levels. The low CO2 levels, the 400 ppm, this is the ambient CO2 levels, and we had an enriched CO2 level, 720 ppm. And here we use 2 different weed species. This is the Echinopodium album that you can see over here. For each of these boxes, we have different combination. This is the low temperature, low CO2, low temperature, high CO2, and as you can see, high temperature, low CO2, and high temperature, high CO2. Now, the treated leaf are marked with T, and the control plants are marked with C. The recommended filter that we use is something that was labeled for California at the moment when I was there in 2019. And you can see here in the number that we have on the right part of the figure or the picture, the amount of survivors, the percentage of survivors. Now we can see that when we increase the CO2 level, we increase the amount of survivors. Plants look better than in the first treatment, low temperature, low CO2. But when we go to the high temperature, we can see that the difference is higher. We see 34% survivors compared to 8% survivors. When we combine, those 2 different parameters, we can see that the number of survivors, the percentage of survivor is increasing. And this means that there is an additive effect of these 2 parameters when they are combined together. When you grow plants under high temperature and high CO2 levels, the herbicide efficacy would be lower than when you grow them under each of these parameters as a single parameter. The same thing was shown for Coniza canadensis, although the differences visually were less seen. Of course, Coniza canadensis is a rosette leaf plant, and this is an erect plant, so there is a difference in architecture. This is why probably we can't see the same response, but still, we can see that the elevation in the number of survivor, percentage of survivor, was pretty much the same under the combination of both high CO2 and high temperatures. After that, we looked at glyphosate translocation under the temperature and CO2 combination. This is something that is more related to the mechanism of this phenomenon. What we did is the whole way experiments, translocation, absorption. We looked at the amount of glyphosate in the C14 glyphosate in shoot, meristem, and undeveloped leaf. These are the young parts of the plant. These are the parts that has the most amount of glyphosate moving to them. This is the big source of the plant. And we looked at them also at the roots. The response that we saw for Chenopodium album and Coniza, as you can see over here, the dashed line is the low temperature, sorry, is the high temperature, high CO2, and the solid line is the low temperature, low CO2. On the y-axis, we can see the C14 compound percentage out of the applied amount. And on the x-axis, we can see the time of the experiment. And this is what we saw generally for these two different parts of the plant. When we looked at high temperature and high CO2 level, we saw that there is an increase in the amount of glyphosate in those specific parts. And this is something that was very interesting because as we saw, those plants have survived the treatment. So if we see that there is those plants survive the treatment, how is it the fact that we see that there is more of the herbicide moving to the shoot, meristem, and undeveloped leaves and the roots? Like, this is something that is very interesting. The other thing that is interesting is the time that it takes for the herbicide to move. For an example, only 24 hours after application, we can see that there is a lot of herbicide already in the high temperature and high CO2 compared to the low temperature and low CO2. Now, the phenotype that we saw and the translocation pattern of glyphosate that I showed you over here made us think about the mechanism of resistance, why these plants are not being injured by herbicides as the plants that are grown under low temperature and low CO2. And we came up with a model that can explain the climate change related reduced sensitivity. Let's say, for an example, that you have this very nice plant. This is a Chenopodium plant, and you spray it with herbicide, with glyphosate. The first day of application, this is how it looks like. 48 hours after application, there is a very vast translocation to the young meristem and to the young leaves, also to the roots. As you can see, the red marks the herbicide, the translocation pattern. The rest of the plant actually doesn't have a lot of herbicide in it. What we see afterwards is we see that the leaves, the young leaves and the meristems look like they are injured very severely, look like they are controlled, but after 14 days, we saw some kind of an overgrown of the plant from secondary buds that are below the initial part that was controlled by the herbicide. Now, this pathway of distribution through the plant can suggest that when glyphosate is moving very fast to these areas, to these strong sinks, it can impair its translocation out of the strong sink, and this is why it cannot move and damage the rest of the plant. So the actual concentration of the herbicide in the lower part of the shoot is very low, and this is why you can see the initiation of this sprouting and the regeneration of the plant. If we look at the plants 70 days after application, this is what you see over here. You can see the phenotype that I was talking about. Instead of one erect plant, as you can see for the control plant, we have a very bushy plant with a lot of stems coming from the lower part. For Coniza canadensis, the phenotype was even more severe. We haven't seen any flowering 70 days after application, but we saw a lot of shoots coming from this area. We believe that the whole mechanism is also related to the loss of apical dominancy, and the herbicide, as I said, is actually impairing its own translocation. And this is something that is very interesting. We didn't have the chance to further investigate it, but I think that there is a lot of work that can be done in order to explain or further explain this mechanism. So to conclude this part, I can say that reduced herbicide sensitivity in response to increased temperatures and CO2 levels and the combination, this is something that we saw in a lot of studies. There is altered herbicide metabolism, absorption, translocation. This is the basic pathways that we see in non-target site resistance. And this is something that is important to remember. These are the mechanisms that are parallel to non-target site resistance. So the response to climate change, for what we see in most of the studies, is parallel to non-target site resistance. Even at the same species, there can be a different response. And we saw that In some species, the glyphosate will affect under low temperature. In some, the reduced sensitivity is going to be under high temperatures, and it's really related to the herbicide and also to the species. Now, this is a very interesting question that I asked myself for many times, and I also spoke to a couple of colleagues of mine, and we tried to think about it and discuss it more thoroughly. Can we actually predict what's going to happen in conditional resistance? Is the knowledge on the evolution of herbicide resistance can be used in this one? For an example, if we know that there is a species that tends to gain more herbicide resistance, or the evolution of herbicide resistance is faster in this species, can we also say that under climate change it will have reduced herbicide efficacy? And this is something that is very interesting to think about. I didn't do a meta-analysis to try and compare those things, but you know, if either of you wants to work on it, I think it can be a nice subject to a master thesis or even a PhD work. Okay, so let's do a small recess, and if you have any questions, I'm going to open up the screen for a second. If either of you wants to raise his hand or ask something, please feel free. You can open your cameras, your microphones. I can see that there are a lot of students here, right? Because of the black boxes, students tend to not open their cameras.
That's true. I think Diana has a question there, Maor. It's in the chat.
Okay.
She has several questions, actually.
Oh, okay. Now I saw. Now I saw. Okay, Diana, do you want to open your microphone and ask me the question? Because this is, it can be like, you know, easier from instead of me reading the whole thing.
Hi.
Hi.
So I read the question. At first, congratulations for the seminar and the work in your lab. I read a lot of articles of you. I love the concept of conditional resistance, and I had a lot of doubts before the seminar. So now would be, I want to ask about, for example, efficacy about herbicides, but thinking about market, you know, companies should have to do again evaluations about sensitivity of herbicides in the future scenario about climate change, you know?
Yes, yes, you're right. You're completely right. I think this is a very, very important point, because when we look at those herbicides today, we had all the trials for herbicide efficacy for a long time. For example, pinoxidine or dichlorphosmethyl or ALS inhibitors that we use, we had a lot of experience in these companies that showed that it's effective for these species, for these crops. It can reduce herbicide, it can reduce weed damage. But now when climate is changing, there should be a reevaluation of herbicide efficacy according to the climate change. And this is an important point. We are really trying to, you know, show the data also to the companies. And I have spoke to some of the people in Bayer and also in other companies like BASF to try and, you know, reassess these issues in the field level. I think that the main issue should be field experiments that are targeted in order to find out those differences in herbicide efficacy. For an example, if I would want to produce this kind of a field experiment, then I'm gonna try and spray my weeds in the field when I know that 2 days after that, there's gonna be an extreme weather event, okay? And compare it to an experiment where I did the same thing, where the weather was the same. And then try and see exactly like you said, what's gonna be the response and reevaluate the efficacy of the herbicide. One other thing that you can already see for us in Israel, actually, you can see that in some of the labels of the herbicides, they have placed some kind of a warning. For an example, if there is an extreme weather event of over 25 degrees, you should not spray the herbicide. Or if there are going to be 2 hot days after herbicide application, you should not spray the herbicide, you should wait for at least 2 more days. And this is something that we are trying, we are trying to get it into the labels, but it's something that should be further investigated, yes.
Okay, thanks. So about the protocols to evaluate resistance to herbicides now, we have followed with like a common path, So what is the common path to evaluate and confirm conditional resistance now?
Mm-hmm. Yes, so this is something that should be done in, climate change facilities. For an example, the work that Louis Ziska did was done in climate change chambers and also with a system that's called FACE. This is a system for CO2 enrichment in the field. And he did a lot of studies using those systems. If you have, those systems, you can actually mimic, for example, 2 different conditions in 2 different chambers, grow the plants at the same time, spray them at the same time, and then look at the response. This is actually part of the work that I did during my PhD and my postdoc, and we are doing also today in my lab. So you have to have like very sophisticated, this is a technical issue, this is something that you get to this point with a lot of people that are working on climate change. They would probably say, okay, I need to have a very, very high-tech facility in order to test for this specific response. And they are right, it's very complicated. But if you have them, this is the way to try and measure the effect of climate change.
Okay, thanks. And just 2 questions, can I do?
You can feel free to ask as many questions as you want.
Well, about tolerance in crops, what do you think about could happen about tolerance or selectivity in the future with crops, thinking about climate change? And the second question is about after seminar and read your papers, I have the idea that conditional resistance is more about physiological process, but the works could be made in conditions, for example, about hours, then I can conclude something about physiology. But what do you think about epigenetic or selection pressure about the future in thinking in conditional resistance?
So for the first question that you ask, I think that there are a lot of studies.
Selectivity in the crops.
Yeah, you ask about crops. Yeah, you ask about crop selectivity, and I think that this is a really big subject. Like there are a lot of papers that are discussing this, and breeders are actually now going back to the wild species in order to get those specific traits that can unlock these kind of advantages for the crop. For an example, if I'm comparing domesticated wheat versus wild wheat, I can see that the wild wheat has drought resilience that the domesticated wheat doesn't have, because it was bred for many centuries for specific yield characters. So now the breeders are going back, trying to get some of these characters back into the genome of the domesticated wheat. And I think that it's a huge subject, and I'm sure that you have a lot of and a lot of reading material that you can find a lot of research in it. For the second part that you talked about, if we talk specifically about the physiological and the epigenetic or genetic mechanism, wait a second with that, because this is the next part of my presentation.
Okay.
So I'm gonna answer that. Okay.
Thanks a lot.
And if not, you can ask again. Okay.
No more questions.
Okay.
No. Thanks a lot.
Sure. Any other questions? Anybody else wants to ask us a question or something? Okay, I haven't seen anything new in the chat, so I'm gonna share my slides again, and I'm just gonna proceed from the same place. Okay, so if you have any questions, of course, feel free. You know, the chat is open and you can ask whatever you say. Okay, so the next part is going to talk about stress-induced herbicide resistance. And this is the part where I'm also going to try to talk about the mechanism, but also on the phenomenon itself. Because when we look at this stress-induced herbicide resistance, we know that there is some kind of a transgenerational communication between plant species. For an example, if a plant species will grow in a hostile environment, an environment that has high drought ratios or low water availability, it would probably some kind of do decoding to its genome so that his progenies will know how to cope with this environment. And this is the transgenerational communication that we have in plants. There is a relationship for the stress-induced herbicide resistance that I'm going to show in a second. But first, let's talk a little bit about transgenerational communication. Now, we all know that, you know, when you have different animals in the animal kingdom, we have communication between parents and offsprings, and they can teach the offspring how to hunt, they can teach them how to communicate between each other. And the fact that these two generations are living at the same time, it means that they can communicate between them. Sometimes the communication is not that good, and you can say that, you know, some people are not, or some animals are not communicating well to their offsprings, and that can affect their fitness, their ecological fitness. If an offspring does not learn how to hunt or how to communicate with other members of the pack, it would probably not survive. So this is something that is very important for the survival of different animals and other organisms. But plants do not interact at the same time. So they have to somehow communicate the stress, as I said, to their offspring. There can be an environmental stress. We know other stresses from water stress. We know also salt stress. We know that there can be stuff that are related also to specific predatory effects. We know that some of the plants can produce allelochemicals that will reduce the competition between them to other plants, or some kind of chemicals that will reduce the herbivory effect that we have. For herbicides, we know mainly about sublethal herbicide exposure, and I'm going to talk a little bit further about this specific thing. I'm going to show some examples, but in general, we know that if we expose the plant to a sublethal dose of the herbicide, a low dose of the herbicide for several generations, we can reduce the sensitivity of the specific population to this herbicide. This is some kind of programming that we're doing to these plants using low herbicide rates in order to increase their resistance or decrease their sensitivity. Now, these transgenerational effects can be something that is for 1 or 2 generations. Most of the time, it wouldn't last forever because it is mainly epigenetic. Genetic changes on transgenerational effects are very rare because mutation that will cause resistance in the next generation is very rare. This is something that is a long evolutionary process, and it mostly don't happen in 1 or 2 generations. So this is why we are looking more on epigenetic effects. Now, for the first example, and this is something, you know, there are a lot of studies related to the response to low-dose herbicide selection, and here I brought 2 studies. The first study on the left side is showing 2 different weed species, Avena fatua and Lolium rigidum, and we can see the survival percentage and the dichlorphos methyl level. And what we see is in the solid line, it's the parental population, and in the dashed line is the progeny population. And we can see that the progeny population is less sensitive to the herbicide than the parental population. And this is after 3 or 4 selection cycles. That means that most of these studies are related to 3 or 4 selection cycles. You can see also on the right side, another study that is related to Lolium rigidum, and this is a different herbicide, peroxasulfone, very low chain fatty acid inhibitor. And we can see the plant survival. And there is an increase when we go further with generations until the dotted one over here. This is the 3rd generation of selection compared to the parental generation, parental population. You can see here very nicely that it works. We also did it with some other herbicides, but it doesn't work with all of them. And I'm not showing this research over here, but we did it with glufosinate a couple of years ago when I was in Davis. We tried to select Lolium rigidum population for glufosinate for 3 generations and see if there is reduced efficacy. And we saw a very slight reduced efficacy, but not something like you see over here. Here it's something like 2 times or 3 times reduced efficacy. And we had something that was very small. But so there is also some kind of a connection between the resistant mechanism that is predominantly in this species to the fact if it can somehow produce a memory for its, a transgeneration effect for its offsprings. Another transgeneration effect that we had tested before was to water stress, and this experiment was done when I was a postdoc at UC Davis, and here you can see we tried to do this experiment with Palmer amaranth. We have here Palmer amaranth plants that grown under well-watered conditions. You can see the height of this plant, and some of them grown under water deficit conditions. Now, in this greenhouse, we had 200 plants, 100 were grown under low water conditions and 100 under well-watered conditions. And we randomized them and tested the effect on all the plants. We measured a lot of different parameters, the height of the plant and when it will flower, the anthesis day, and all sorts of other issues, relative growth rate. But after that, we produced 2 offspring population, one from plants that were grown under well-watered condition and one from plants that were grown under water deficit condition. We took those plants and segregated them to 2 different greenhouses under the same conditions in order to not have any cross-pollination between the well-watered and the water deficit condition. And then we tried to test what would be the response of these plants at the offspring population level. And this is what you see over here. We did some seed characterization of the Palmer amaranthus progeny population. The first thing that you can see, the well-watered is WW, the WD is water deficiency. This is total germination. We had 2 populations. We did this experiment with 2 different populations, one from California and one from Kansas. And the first thing that you can see is when we test for total germination percentage, there is a reduction in total germination percentage for the well-watered produced seed. So they germinate less. When we look at the weight of 1,000 seeds, we can also see that the well-watered seeds are, the weight of the well-watered seeds is less than the weight of the water deficiency seeds, which means that they are lighter. We haven't really tested the number of seeds, but the fact is we saw a significant difference in the weight and in the total germination. After that, we tried to see what would be the germination percentage under different water deficiency or water potentials. Now, what we see over here is in red, we see plants or seeds from plants that were grown under water deficit. And in blue, we can see seeds of plants that were grown under well-watered conditions. As I said before, we had 2 populations. These were 2 maternal populations that we grew for the 2 seed populations. And we have different water potentials from -0.8 until 0. And you can see the accumulation of germination. Now, there is a big difference between the well-watered seeds and the water deficit seeds. As you can see, the water deficit seeds tend to germinate better at all water potentials. For the Kansas population, we had higher germination than from the California population, but still the difference between well-watered produced seeds and water deficit produced seeds is very high. The germination of water deficit seeds is higher. They have some kind of a memory of the water deficiency situation that the maternal plant was in, and they can cope with it better than the plants that were grown under well-watered conditions. And this was something that was very nice to see. If you want to see the paper, you can look at it. This is for 30 degrees. We had some other experiments for 20 degrees when we saw approximately the same differences, but overall it seems like the same trend. Now, after that, we wanted to see, okay, we saw that there is a difference. We knew that these plants suffered from water stress, and we saw that they have some kind of advantage under water stress. But what's going to happen if we will spray those plants with herbicides? Will they also have an advantage under herbicide application? Can one stress, water stress, cause resilience to other stress, herbicide stress? And in here, we try to look at, as you can see, 5 different herbicides from different mode of actions, auxin inhibitors, and metolachlor is VSCFA, and rimsulfuron is ALS, and we have sulfenacil, the PPO, and we also have the simazine, PS2, still the 2 populations that I showed you before, and seeds that were produced under water deficit and well-watered condition. Now, this is the visual injury under different herbicide application rates. Now, the highest rates that we used was, I must say, was half of the recommended rate. We didn't want to use the full rate. And still you can see that there is a difference between the response of plants that were grown under water deficits and produce seeds. These seeds tend to be less sensitive to the herbicide. When we look at rimsulfuron, we can see that there is a difference that is very significant between those 2 seed lots. And also when we look at simazine, the PS2 inhibitor, so plants that were grown under water deficit can produce seeds that are more resistant or less sensitive to simazine and rimsulfuron. Now, I'm sure that if we will assess these kind of things in other population, in other weed species, we would probably see a different response to different herbicides, but I think that the general trade will stay approximately like this. Now, when we think about these mechanisms that are behind this phenomenon, we know that there are all sorts of epigenetic responses that can happen in plants that are grown under stress. Either it's differences in methylation or the chromatin, the expression of those specific areas in the genome. These epigenetic processes or these epigenetic alteration can happen in 1 generation. They can actually exacerbate in 2 or 3 generations under stress that is subsequent. For 3 or 4 generations, we can have more of these responses. But this is one of the cases that I found. There is, in my surprise, there is only 2 cases that have studies, stuff that are related to methylation alternation or chromatin alternation in response to herbicide injury. This specific case is a paper that was published not a long time ago in 2021, and here you can see differences in methylation under the rate of glyphosate, 5% or 10% of glyphosate. They have used sublethal doses of glyphosate in order to provoke the different methylation. They didn't want to control the plants, and this is why they used low rates of glyphosate. They collected the seeds, and then they tested for differences in methylation. And this is what you see over here. As you can see, there are differences in methylation between the different treatments. For an example, we can see that there is a reduction in methylated cysteines in 10% rate of glyphosate. There is also a difference between the sequence context, as you can see over here, but the differences are not very high. But when we look at the GC content that you see over here, differential methylation of as a percentage of the methylated cysteines, you can see that the chromosome glyphosate rate, this is the chromosomes, the different chromosomes of Arabidopsis thaliana, and you can see the change of different chromosomes is pretty high. Like if we look on the difference between 5% of glyphosate rate and 10% glyphosate rate, we can see that the chromosome change is almost double than what we see in 10 than what we see in 5%. And this is something that was very interesting. We have here also hypermethylation and hypomethylation. we have a change in the number of sites that have more methylation, and also a change in the number of sites that has less methylation. And changes that are related to methylation can open those different areas that will somehow bring into expression genes that are more related to herbicide detoxification in the progeny generation. And this is something that is very similar to what we see in related to other stress, such as water stress or salt stress, when we see that the progeny generation will be more resistant, and the epigenetic shows that the areas that are related to that are related to genes that are part of the detoxification pattern or part of tolerancy pattern for different stress, abiotic and biotic stress factors. Another research that was previously published in the last year, and this is from a group from Brazil, in the International Journal of Molecular Science. And here, those researchers have tried to look at different Arabidopsis thaliana chromatin mutants. They were looking on 3 different herbicides compared to control, and we're trying to see what's going to be the response of these chromatin mutants to the herbicides. Now, if we're looking on chromatin mutants, we know that those specific areas are either open or closed, and they are the difference in they may be the reason for the difference in herbicide response of those specific mutants. And as you can see, for example, the RST1 has a very good response for the herbicides. And we can see that even when plants are sprayed with 2,4-D, the response is very different from other mutants. There were others that were less sensitive to the herbicide, such as the NRPPD2A, and it was most likely related to the chromatin opening on those specific areas. Now, in order to try and look further into this issue, of course, we have to create our own mutants. We have to see what's going to be the response to the herbicide, which are the genes that have been expressed in the process of chromatin opening in those areas. Some of the genes that have been expressed and people have shown before are related to all sorts of sequestration processes. If we talk about MRP or other ABC transporters that were also part of this, and these would be the genes that we would be looking for in those specific open chromatin areas in order to somehow explain the response for glyphosate or for other herbicides that are related to this detoxification pattern. So there was not a lot of data on these things, and as you can see, only 2 papers. So whom of you who will decide to work on things that are related to chromatin or all sorts of, epigenetic response to herbicides or to climate change induced response to herbicide, I think that can be a very nice, PhD or master thesis. So I encourage you to try and take these subjects and work on that in the future. So to conclude this part, I'm going to say a couple of things about it, just a couple of points that I want you to remember. First of all, we see that sublethal herbicide does cause multiple plant responses that can result in non-target site resistance. And this is something that we see in different studies. And the mechanisms that are related to that are probably the same as mechanisms of non-target site resistance for different herbicides. There's not a lot of difference between them. Maternal plants that are subjected to environmental stress may epigenetically modify certain phenotypes creating their progeny. We saw how plants that have been exposed to water stress produce progenies that are more tolerant to water stress and also more tolerant to herbicide application. So the herbicide sensitivity is reduced, and the water or the drought sensitivity or resilient is also increased. So these are the things that we should be looking for. When we look on the big picture, plants that we are trying to extinct under, or weeds that we are trying to extinct or control under climate change, may gain these kind of characteristics that would make it more difficult for us to control them. And this is why we have to study those issues and learn more about the response of weeds to this transgenerational effect. Now, as I said, most of these changes are some kind of soft inheritance they will be there for a generation or two. But if the stress is removed from the environment, for example, if those plants in further progeny generations will grow under well-watered condition, they will most probably lose their ability to be tolerant to those stress, to the water stress. What will happen if those plants will grow under well-watered condition? What will happen to their herbicide resistance or herbicide resistance sensitivity? That's another question that we don't have the answer to, and also another thing that will be very interesting to study in the future. Epigenetic are less stable, as I said, and we have to understand more about them in order to know how to cope with them. So at this point, I'm going to move to the third part, and this is actually the last part of my presentation. And here I want to talk a little bit about approaches that should be considered in order to manage conditional resistance. Okay, if we look at conditional resistance, how we can prevent it, how we can cope with it, and then we most probably talk about how we can prevent herbicide detoxification, how we can cope with herbicide detoxification, how we can reverse this specific response to herbicide. If we look at a plant at the moment and we see that it is tolerant to the herbicide and it has a non-target site resistant mechanism, what should we do in order to cope with that? Now, there were several cases in the past, several studies that were working on reversing, and I'm saying reversing, you know, it's not the real word to say here. It may be, you know, preventing detoxification is more suitable here, but they have done some studies that are related to how to reverse the effect of the herbicide. So here you can see in the figure, one resistant and one sensitive population of Apera spicaventi. These populations are resistant to ACCS inhibitors, ALS inhibitors, and you can see the response to different herbicides. And what the authors of this paper did, very excellent researchers from Czech Republic, they tried to reverse the activity of the herbicide or the response to the herbicide using malathion. Now, malathion is a pesticide. It's an insecticide. It's been banned in most of the world because this is a phosphoric compound, a very dangerous chemical for humans and also for other organisms, but it was used to control insects. The other part that it had, or the other effect that it has, is it would probably reduce the activity of cytochrome P450 enzymes or other detoxification enzymes. And what we see over here is the use of malathion in order to reverse the resistance. This is actually part of a very systematic practice that we do today in order to somehow detect the mechanism of resistance. We use malathion as some kind of an indicator for non-target site resistance that is related to detoxification of the herbicide. Here you can see there is the dry weight on the y-axis, and on the x-axis you can see the different herbicides that they use. And you can see the combinations of resistant with malathion, the sensitive population with malathion, and resistant and sensitive alone. Now, in almost all cases, we look at the sensitive population, we can see that it was controlled very nicely, and malathion did not really change the picture that we saw. When we look at the resistant population here, it also didn't change anything, because peroxasulam, probably the mechanism of detoxification as peroxasulam is not related to enzymes that the malathion is inhibiting. The same thing we saw for chlorotoluene. It's also an ALS inhibitor. Probably the detoxification is not coming from enzymes that are related to malathion. But for pinoxidine, we can see that the sensitive population acts almost the same with or without. But within the resistant population, we can see that the resistant almost disappears when we combine malathion to the treatment. Now, this is something that it's very interesting to say because it means that we can reverse the resistance when we can actually show that the mechanism of resistance is related to detoxification enzymes, probably cytochrome P450 enzymes, as has been shown in other studies. Now, there is another detoxification process that all of us know, and it's related to GST, glutathione S transferase enzymes. And one of the chemicals that is being used to reverse this is NBD chlorine. Here we can see treatments with NBD chlorine combined with quizalofop methyl and ACCase inhibitors, malathion combined with quizalofop and quizalofop alone for two populations, one sensitive and one resistance of polypogon fugax. We can see that there is a different response when we look at the resistant population with NBDCl plus quizalofop. We can see that the RI, the resistant index, is 1.2. When we look at it with malathion, we can see that the resistant index is pretty high, almost as high as with quizalofop. The same thing was shown for the sensitive population. We see that the mixture with NBD or with malathion doesn't change the resistant index. Those researchers that examined that showed that the actual mechanism of the response to GST is very interesting. Like, they show that there is GST activity for the resistant population. As you can see over here, days after herbicide application, we can see the GST accumulating in the resistant population. And in the sensitive population, we can see that it takes more time. As I told you before, in the detoxification processes, the time has a very, very important point. If you can detoxificate the herbicide fast enough, you will survive. If you cannot, you will probably be controlled by the herbicide. In here, we can see also that when we look at the GST relative RNA expression, it's higher in the resistant population than in the sensitive one. There are different GSTs here. The GST2 and GSTL3 are higher, and the GSTL1 is lower. So we can assume that these two enzymes or two genes are related to the mechanism of resistance. So we learned here that we have also NBD. We don't only have malathion, but also NBD. And NBD is mostly related to detoxification processes that involve GST enzymes. There are another studies. There is another study from Australia that was also showing how malathion can be involved in resistant mechanism, non-target site resistance. In here, it's for diflufenican, fluoridone, HPD inhibitors, herbicide that works in a different mode of action. And you can see that when they sprayed those plants, the Raphanus plants, the wild radish plants, with malathion alone, there is no response. It looks like the control. When they sprayed it with diflufenican or fluoridone alone, you can see that there is some kind of an injury, more or less. They are less sensitive for the fluoridone, but they are more sensitive for diflufenican. But when we combine the malathion with diflufenican and fluoridone, we can see that the response is very severe, which means that part of the mechanism of resistance or reduced sensitivity that we see here is related to cytochrome P450 enzymes. It doesn't always work. Some of the herbicides are not really working in these mechanisms. And here you can see a very nice study that was done. This is an Amaranthus tuberculatus population that has 6-way resistant to many herbicides from different mode of actions, 2,4-D, atrazine, we have mesotrione, chloromiron, a lot of herbicides from different mode of actions. And you can see the response to the herbicide combined with malathion and without malathion, and with NBD or without NBD. And this is a clue for the resistant mechanism. For example, if you take 2,4-D and combine it with malathion, we can see that there is a reduced shoot dry weight. It means that malathion can probably inhibit some of the enzymes that are related to detoxification of 2,4-D. When we look at mesotrione, we see the same picture. There is a reduction with malathion. The same thing for chlorimuron, an ALS inhibitor. When we look in atrazine, for an example, we see that for both malathion and NBD, we don't see any response. So it can say two things. Either the enzymes that are working for detoxification are not related to these two, are not being inhibited by these two compounds, or they have target site resistance. And this is why they don't see any difference when they spray it with atrazine and malathion and atrazine with NBD. And this was actually part of the research. You can read more about it and see the difference between the target site and non-target site resistances that the researchers have found. After I said all that, and we talked a little bit about malathion, we talked about detoxification and how does it work, I want to show you how does it work in a background of temperatures. Like now we throw another parameter into the equation. We are not only looking on the combination between malathion and the herbicide, we are also looking on the change within temperatures. I want to remind you that we saw before that under low temperature, our brachypodium accession that I showed you before is very sensitive to pinoxidant, to an ACCase inhibitor. And under high temperature, it would be probably more resistant or less sensitive to the herbicide. Here we did an experiment when we did a lot of treatments with malathion and pinoxidant, half of the recommended dose, malathion plus pinoxidant, half of the recommended dose, pinoxidant alone, and malathion with pinoxidant, the full dose for both of them. And we try to see under different temperatures how the weed will react to this combination. You can see the increase in temperature from 1016 to 2834. And you can see very nicely, this is the sensitive biotype and this is the resistant biotype. You can see very nicely the difference between the two biotypes. If we look at the increase in temperature, we can see that half the recommended dose, we can already see some of the plants from the sensitive and both the resistant biotype that can survive half of the recommended dose. But when we apply malathion with half of the recommended dose, no plants can survive. When we increase the temperature, we can see that the survival increases even with malathion. Like malathion with half of the recommended dose of pinoxidant, you can see some of the plants for the resistant biotype can survive. But for the sensitive one, we don't see any survival beyond half of the recommended field rate. For the highest temperature, we saw that some of the plants survived even a full dose of pinoxidant with the combination of malathion. Now, this figure or this experiment tells us that the response to the herbicide that is related to climate change is quantitative. And it actually reinforces the fact that detoxification processes that are related to enzyme efficacy and that can be part of the temperature changes are part of the resistant mechanism. Because as long as we increase the temperature, there is less effect of both malathion and pinoxidant. And it means that our detoxification processes are more effective. They can work even under even under the high temperature and the part that has the malathion. So this is something that, for me, it showed very nicely how temperature shifts or extreme weather event can be very significant in increasing detoxification and reducing herbicide sensitivity in different weed species. We have other ways that we can improve herbicide efficacy under climate change. These are some of the points that I just want to show you. You can look at different papers that will show some of the new advanced technologies in the world for the use of molecules that can increase herbicide translocation, all sorts of adjuvants that we have in our formulations. Herbicide encapsulation is something that is very important. We see it mainly in human drugs. Today, this industry of encapsulation is very, very important. High-tech technology that can get the specific drug to the specific organ or specific cell without being detoxificated or without being, sequestrated in the plant or in the human body. We can use nanoparticles to reduce functionality, and we can also try to selectively, to work on the fact that we don't want to harm the non-target organisms. That we can use those issues also for this. We can use the stuff that are related to encapsulation and nanoparticles also for this kind of activities. If we look at the application of nanotechnology in pesticide delivery, this is a very nice illustration of how does it work. We have the polymeric particle and the biocide, whichever herbicide that you want to put in, or a bioherbicide or a biocide that you want to put in. You do the encapsulation process, and then that specific, capsule can move through the plant and reach a specific area. For an example, it can reach the chloroplast, it can reach the part that is related to the upper leaf, it can reach the root, it can get to the point where it gained the most efficient way to get to that specific part or organ or cell of the plant without being detoxificated or impaired in the way to do its action. There are a lot of papers on that. I don't want to go into it because it can be a different lecture, a whole new talk, only to talk about nanoparticles and other things. If you're interested in that, I recommend you to try and look for nanoparticles and other encapsulation articles in the issue that can be very interested to whom of you are interested in that. So here is the part where I want to conclude my talk, and this is actually the last slide of conclusion. I want to leave you with something to take home. I want to leave you with some messages that I want you to remember when we talk on climate change and herbicide response and this whole weed management and weed control issues that we're going to face in the future. So we know that climate change will most likely exacerbate the damage, and mainly through the reduced herbicide efficacy. Conditional resistance is something that we should try and look for when we see resistant cases. For example, we had a lot of reports from farmers that said last year we had a very, let's say, hot winter. And a lot of farmers said, okay, I have a problem in my field. I have Phalaris paradoxa that I can't control. I have Lolium rigidum that I can't control. And the field instructors came to me and said, we have a lot of issues with this weed. We collected seed. We want you to test them for resistance. Suddenly, this year, we have a very cold winter. And field instructors are coming to me and saying, this, in the same field that we had problems with herbicide application and weed management, this year, herbicide worked perfectly. So the only difference that I can see between the 2 years, because they use the same herbicide, is the temperature. We had a very warm winter last year and a very cold winter this one. So I urge you to look at the temperature and other environmental factors whenever you come across a herbicide-resistant case before you go and look for the herbicide mechanism itself, because this is something that is very important and can help you. It's proposed that also the mechanisms that are leading for non-target site resistant can play a role in conditional resistance. We saw the impaired translocation, we saw the detoxification, we saw a lot of similarities between these two. And we can probably try and predict which weeds are likely to be less responsive under climate change if we create some kind of a meta-analysis that will show which weeds are more prone to evolve non-target site resistant in those specific mechanisms. And this risk assessment evaluation can be something that we do in order to prevent any issues of herbicide inactivation or reduced efficacy under the climate change that we are predicting and we are already having in our fields. Also, improved herbicide efficacy. I talked about the nanoparticles, all of the other issues. This is the time to say that if some of you are going to work for the chemical industry, they should remember that some of these factors, as we discussed before, can also affect herbicide resistance. The question that Diana had, the discussion that we did is part of it. And I think that you should take it under consideration in new formulation. You should think about it when you produce the new herbicides and when you design the formulation of the new herbicide. Additional amount of malathion or other chemicals that can inhibit herbicide detoxification or can help with herbicide translocation can sometimes make the difference between proper control and failure of control of that specific weed in the field. So it's worth it to try and develop this kind of systems for the future and formulation for the future. So this part, I want to say thank you for all the collaborators. It's been and still a big project that we're working on for analyzing the differences in climate change for herbicide response. My lab in the VAR, and I want to also invite all of you to come to Israel at 2024. Luis would can say more than I do on this marvelous thing. He was also supposed to visit us, but COVID situation prevented that. But still, we hope to see you all in the WS, the International Weed Science Congress at 2024. And if you have any questions, please feel free to ask. I will stop sharing now so we can ask some questions.
Thank you, Maor. I think it was a wonderful talk, and I think we're gonna need another talk about the nano formulation and to discuss a little bit about this topic as well. Is there any question that you want to ask? Let's see if there is any other question that they have. I have one question that I wrote here. Let me see the question. You talk about malathion and the P450 inhibitor as mostly as a tool to test for metabolism-based resistance. But you mentioned that some of the authors try to use as to cope with resistance. Is there any what kind of problems or that can arise from using this approach for managing resistance? Do you see any adaptation in the future if you start using that to cope for this kind of resistance?
Yes. So there are two key points that should be discussed if we're talking about using malathion as let's say resistance reversing chemical. The first one is that malathion is reversing the cytochrome P450 activity. And if we are going to spray weeds in the field, most probably we're going to also affect the crop because, for example, wheat has the same detoxification system as some of the weeds that we have. So if we are inhibiting detoxification of cytochrome P450, it can also damage the wheat. And I must say we did a small experiment and we saw some response. And it's mainly related to the time of application. Like if wheat is strong enough and large enough, it can actually, stand the combination of malathion and pinoxidine, for example. But this is something that is very fragile. So we have to think about a concentration of malathion or another chemical that will be effective to reverse resistance, but will not damage the crop that we have, because most of the crops are using the same mechanisms. So this is the first thing that I think that comes to mind. The second thing is maybe if we are going to use it, then of course, evolution will happen all the time. And what's going to happen is that maybe most of our weeds will shift to target site resistance or to other resistances related to translocation of the herbicide. So as within every chemical that we have or every herbicide that we have, we should not use it all the time. We should have some kind of a thoughtful use in a specific time. We should think about what we're doing with those combinations and not exaggerate until we break that tool. This is like what we always say, you use the herbicide until you break it and it doesn't work anymore. And this is the problem that I think that can happen if we are doing this as well.
Yes, sir. Diana has a question, if you want to read.
Okay, so Diana is asking, do you think that the cases of metabolic resistance that already exist today could have an epigenetic origin? There is a very interesting paper by William Dyer that was published in Pest Management Science, I think in 2018, if I'm not mistaken. And he's really going through the whole epigenetic things that are related to herbicide resistance. There is another one published by Aldo from Brazil, also Meroto, yes, also in Pest Management Science, that is discussing the epigenetic origin. And I must say that, when you look at these plants and you think about it, if you see that the metabolic resistance is evolving in 3 generations of sublethal herbicide use, it would probably mean that it has an epigenetic origin, because the genetic changes, de novo mutations, or even spontaneous mutation, they need more time in order to develop at the level of the population. They need a lot more pollination between individuals, and they need more individuals. Like the guys that did these experiments used most of the time not more than 100, 200 individuals, and pollination within 100 individual for target site resistance is very rare. So I think that epigenetic can be considered as a main reason for that. Of course, we have to test that. We have to take the population generation and the progenies and look on differences in chromatin and all sorts of methylation patterns between those populations, and then try and see if those chromatin or methylations are in areas that are related to metabolic resistance. Like this would be the way that I think that it should be tested in the future.
Yeah. There is a research on a paper from Davis that they found out that the random mutation are not so random on Arabidopsis. It means that epigenetics may be driving the random mutation. Whenever the epigenetics is affecting the availability of that part of genome to be mutated, that means that it may create a more prone to mutation area in the genome, and that's something that we may consider whenever you use herbicides, is that we are changing that the genome part that is prone to mutation, and it's like maybe an indirect. It's only reading the paper that we have this idea, because of course the paper is not regarding the herbicides, but this is something that we may need to look in long term, that maybe we are the epigenetics is sort of not promoting, but facilitating mutation in a given part of the genome.
Yes, it was recently published. It's just something of the last couple of months. It was.
Yes.
Yeah.
I think it was in December, I guess. Yeah, November or December, and this is very, very unique, and this is something that we may consider as well.
A lot of work, young students. They have a lot of projects in line.
Yeah, but we have so many bright students, so that we need many things. We're gonna happen in a few years.
I hope so. I really hope so. Yeah.
Any other question? I think they don't have any other question. Maor, thank you so much for your time and for your willingness to talk with your student. Everybody here are probably from our lab. We really appreciate your time, and it's good for the students to get to know you, and for me to get to know you as well. And you have a very, very nice group there, and very nice project going on. I really appreciate your